Okay, in this video, we're going to talk a little about the biology of memory, like what it really means to store a long-term memory when a memory isn't some little physical marble-sized ball you can stuff into your head or something. So then what is it exactly? That's what I want to get into here. And let's start with a helpful term that we're going to run into a lot, which is consolidation. So consolidation is a process that transforms like a memory from an initial fragile state into more permanent storage. Really, it means the way we've been talking about long-term memory as if it's a single storage place, that's oversimplified. When we talk about long-term memory, that can include retrieving some information from 5 minutes ago or 2 minutes ago, but that doesn't mean that stuff will be available to you a week from now or a year from now. Whereas in other cases, long-term memory has a much more permanent state, right? Or at least a much longer time scale than a few minutes. So consolidation is what makes some long-term memories last much longer. And this process, by the way, it starts happening within seconds or minutes after an experience. This is particularly clear when someone receives a concussion, for example, like during a car accident or when playing a rough sport. Like when a football player runs a play and gets hit really hard and receives a concussion, then even if the player is totally fine later, they'll never be able to remember the play, the moments just before the hit. And that's because their brain was interrupted before it could start to consolidate that memory. And roughly speaking, the process of consolidation, it generally completes within about 24 hours. So there are ways to encode the memory that make the consolidation more robust, like rehearsal for the information or elaborating on it when it's still in working memory. But the most crucial factor for the relative permanence of a memory is sleep. When we interrupt sleep, we find that memory consolidation is interrupted and in essence, less learning or remembering occurs. Now the question is, how does the brain actually store a memory? And it's tempting to ask, where in the brain the memory goes? But what are we even putting in there? What is it that's quote unquote going into the brain? And to really understand memory, you have to understand that a memory is not an object, but a pattern of neural connections in the brain that, when activated, cause some similar experiences or thoughts or perceptions to what occurred back when you had the original experience you're remembering. Even the word remember comes from re, meaning again, and the Latin memorare, to bring to mind. So we're bringing something to mind again. We're reactivating a thought or experience that's constructed by the brain. So when we ask how the brain stores a memory, the answer is that it's making some lasting changes to a complex network of neurons that we've got up there in the brain, making a particular pattern of neural firing more likely to happen again in the future. In other words, the brain stores a memory by creating new connections or strengthening existing connections between neurons. That's what memory is. When you practice riding a bike, right, you're building the procedural memory for that skill. You're taking advantage of neuroplasticity, of the brain changing its wiring on the fly based on your experience, such that it can repeat the stuff over and over again more easily next time. So each time you ride a bike, there's like a symphony of neural activity happening in your motor cortex at the back of the frontal lobe, also in your basal ganglia, in your cerebellum. These brain areas are coordinating your muscles and getting feedback from your vision and your balance system, doing all this stuff. And as you gain that bike riding skill, you're basically just making that symphony easier to activate and more efficiently wired and better coordinated for the future when you're in that situation balancing on a moving bike again. So you can think of making memories or learning as causing changes in many thousands of synapses that are connecting neurons together. And the memory itself is really that firing pattern across groups of neurons. So if you have a memory of standing in an art gallery, looking at a beautiful mural, if you're thinking back to that time when you were looking at that mural, what's happening during that memory is your visual cortex will be activating much in the same pattern as when you were originally there in person staring at the mural itself. To remember something you saw is to reactivate a similar pattern of firing in your visual cortex. To remember something you heard is basically to reactivate the neural activity that happened when you heard that thing originally. To remember the fact that a cat has 4 legs is to have made some new connections between your neural representation of the concept 4, your neural representation of the concept leg, your neural representation of the letter, shaped like C and A and T, your neural representation of the sound cat, and so on, so that in the future, when you activate some of those neurons, like hearing the sound cat or seeing the letters C, A, and T together, it also co-activates some other activity that represents number of legs. It's really just neural connections and neural activity at the heart of everything we learn, know, and remember. And indeed, the foundation of this very idea goes back to Hebb in the mid-century. He hypothesized that learning and memory are represented by physiological changes at the synapse, where our neurons meet. So he put it this way. He said, let us assume that the persistence or repetition of a reverberatory activity or trace tends to induce lasting cellular changes that add to its stability. In other words, neurons that keep firing in the same way, persistent or repetitive firing, right? That memory trace tends to induce lasting cellular changes, meaning neuron changes, that add to its stability, that firing stability, that memory stability. When an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells, one or both neurons, in other words, such that A's efficiency as one of the cells that fires B, that turns on B, is increased. So that's his way of putting it, right? But nowadays we have a term for this change in the structure and connection of neurons at the synapses. We call it long-term potentiation, or LTP. When an action potential of one cell activates another cell, the synaptic connection between them gets stronger, and or new synapses can be formed. So you'll often hear this phrased as cells that fire together, wire together. So if you keep looking at the same mural every day, you're activating the same basic patterns of activity in your visual cortex over and over again, right? Which means it's going to become easier and easier for that whole cascade of activity to happen again in the future. In other words, you will remember it better because you've activated the same pattern of neurons together multiple times, making it easier for that same pattern to come alive again. If you've only seen the mural once, then it's less likely that later on your brain will be able to activate the exact same pattern of neural firing, and at best your memory of the mural will be limited, right? Maybe limited to the gist of it or some detail that you happen to focus on and attend to for a while. That's the reason some memories are stronger or weaker than others, and some facts come to mind more easily or less easily. It's about how easy it is to get that pattern or a similar pattern of neural firing to happen in your brain again. And a fundamental factor of that is long-term potentiation, like neurons that fire together become more likely to fire together again in the future. Now, we're not going to get into the nitty-gritty of the biochemical basis of long-term potentiation, but you can think of it kind of like this. Where two neurons meet, we've kind of shown here, we've got a terminal button of one axon and a dendrite of the next neuron that receives the information, right? Where they meet, the activation of this connection causes some moment by moment and also some lasting changes in the structure of the two neurons in such a way that the threshold for firing again in the future becomes lower, becomes easier to activate that receiving neuron in the future. And if this happens across a whole network of neurons, like the pattern of many, many neurons that are involved in an experience, like looking at a mural or during your studying of a vocab word over and over again, then we get potentiation of that whole pattern of activity across the network of neurons. So here's just another diagram of the idea, so you can see some of the mechanisms involved in LTP. So from panel 1 to 2 here, you can see the change of adding some more receptors on the dendrite itself, the receiving dendrite on the right, more places to catch those juicy neurotransmitters floating across the gap. But you can also see in panels 3 to 4 here that it can involve the presynaptic neuron, so the one that's initially firing and transmitting some stuff to the next one. That first neuron actually might end up putting out more neurotransmitter. So there are multiple ways that we get these lasting changes at the synaptic level where two meet. That's called synaptic plasticity, synaptic change or flexibility, right? It's actually the most fundamental kind of neuroplasticity in the brain. Now, probably the most important thing to understand about how memories are stored in the brain is that they are not located in any one area. Like long-term memories are distributed largely throughout the cerebral cortex, the lobes of the brain. They are stored in the form of tons of connections between neurons, which can include connecting brain areas that are pretty far apart actually. And it's those changes in the structure and the connection of synapses that are a neural record of our experiences. That's memory. And this often confuses people who have just learned a little bit about the brain, because you may have been taught that the hippocampus is the, quote unquote memory area of the brain. So people often think memories reside in the hippocampus, and that is just not true. Although the hippocampus does have a really important role in some types of memory. So specifically, the hippocampus is really important for consolidation. It helps kind of coordinate and reactivate those distributed connections and contexts, especially while you sleep. In some sense, the hippocampus is like a conductor that replays the symphony of neural activity throughout the cortex that was associated with the original event. But after consolidation happens, after the memory is laid down in a more lasting and permanent state that's kind of distributed throughout the cortex, then the hippocampus is no longer needed. So for example, if we use fMRI to scan brain activity during retrieval of memories, so you just put people in a scanner and you have them do some memory task, right? And they think about some memories. We find that a lot of hippocampal activity for recent memories shows up. So if you're asking them about something that hasn't had a chance to be consolidated yet, something they just learned or had an experience 10 minutes ago, we get lots of hippocampal activity. So activity in the hippocampus when they're remembering that. The red bar for hippocampus activity is high here, right? But for memories that are older, ones that you've already had a time to consolidate and you've slept on, when we go back and remember those things later, it involves basically no hippocampal activity, as you can see on the right here. Rather, we see a pattern of neurons across the cortex firing when we remember those consolidated things. So here's a way to think of it. On the left in the panel A here, we've got soon after a memory began forming, the hippocampus is doing some work. It's activating all these connections between different areas in the cortex that were involved in that experience, right? There might be visual parts of it and auditory parts of it and maybe words and thoughts that were activated during that time. But as time goes on, we get less and less firing between the hippocampus and those cortical neurons out in the lobes of the brain, out in the cortex. But more and more of those cortical neurons are connecting to each other and co-firing, because cells that fire together wire together, right? And the hippocampus makes sure those cells fire together over and over again over that initial 24 hours or so after the experience. So this whole idea can help us see that long-term memory is not a binary yes-no or on-off kind of process. Long-term memory, in the sense of being anything of duration longer than working memory's 15 to 30 seconds, well, that can include being able to remember some details from the passage you read 10 minutes ago, sure. But if we don't focus on those details, if we don't practice recalling them, if we don't connect them to other concepts or encode them in some other way that increases consolidation, then they're going to be relatively temporary, even though they're quote-unquote long-term memory. But the more we let consolidation happen or help it happen better, the more we get a lasting pattern wired into the brain. So long-term memory is kind of a continuum, a spectrum of more or less consolidated, more or less wired in as a reactivatable pattern across the relevant parts of the cortex. That's what memory is. Here's just another diagram of that from a study in 2017. What they did in this one, it was identify when and how the consolidation process is happening, specifically in this case while we're sleeping. So as I alluded to earlier, lasting consolidation of memory really relies on sleeping. And for a long time, people assumed that this consolidation that happens during sleep was mostly going to be just a REM sleep phenomenon. So something that happens during rapid eye movement sleep, during the dreaming part of sleep. Kind of like, oh yeah, I'm reliving my day in my dreams to help set those memories down into a more permanent state. That's a reasonable hypothesis, right? And that would make sense because sometimes our dreams do relate to things we experienced or we're thinking about that past day. However, it turns out a lot of current evidence points toward the hippocampus doing its consolidation work during what we call non-REM or slow wave sleep, sometimes just called deep sleep. So in non-REM, kind of mostly non-dreaming sleep, we get the hippocampus doing a lot of its work. On the diagram here, if you start on the left, the actual experience we had, right, the thing we learned during the day, as we've seen before, the way you encode the information matters for how well it gets into long-term memory. but that's like the yellow areas here going in, right? Information incoming in and activating our sensory and other areas of the cortex during the experience. And then that information flows through to the hippocampus, kind of in blue at the bottom there, if you follow those green arrows down. But then in the middle of the diagram, the two boxes in the middle here, that is what they found where like we're sleeping. So it's nighttime, we're sleeping. What they found is during non-REM, slow wave sleep, the hippocampus is sort of replaying the temporary memory trace, which means it's integrating those connections back into the cortex, which you can see with the upward blue arrows here. Then later, when we're awake again, so on the far right of the diagram now, we can retrieve or reactivate the information without needing the hippocampus. Like you can see with the green arrows coming upward at the top here, right? No hippocampus needed to get the information back out now. We can get it from the cortex itself. And also it can integrate new information into the memory or update it, like with those yellow downward arrows. So that's the rough idea of how consolidation is happening and how sleep is important for that. And it's definitely not just a matter of replaying your day in your dreams. Now, the natural next question would be, if a memory is consolidated, is it permanent and immutable? And already we can see from the far right side of this diagram here that probably isn't the case. Those yellow areas moving downward and more green arrows moving downward mean that new experiences might update or change our original memory. And in fact, there's a lot of other work suggesting that consolidation is a little less permanent, a little less immutable than we'd like to think. Because it turns out retrieving a memory can actually make it fragile again. So we have to go through a reconsolidation process. We have to consolidate again after we've activated a memory, or else it might weaken. And this is weird and kind of counterintuitive, because you would think that activating a memory, like thinking back to what happened yesterday morning, thinking back to that time I was in the art gallery looking at the mural, you would think that would, if anything, just make the memory stronger and more secure, because that's long-term potentiation, right? But remember, our system also has this hippocampal process of consolidating things, kind of consolidation, throwing a wrench into things. So each time you remember, say, an episodic event from long-term memory, you are strengthening it. That is true. But you may also end up altering the memory each time. Kind of like how some other neurons might end up getting connected that weren't before just because they happen to be firing during this memory, right? You're in a different place, you're in a different mood, different inner state. That might somewhat get wired in with the rest of that pattern. In fact, we can see this in a simple cognitive experiment. If we just randomly assign people to two groups, an experimental group and a control group, both of them lay down some new episodic memory. It's identical for people in both groups. But with the experimental group, we have them recall that memory multiple times, while the control group does not. They don't actually go back and retrieve that memory until the very end. Then at the very end, what we do with both groups, we go back and we test them on that memory. The experimental group, they retrieved it multiple times along the way. Turns out they have more errors in their memory, like more false details end up added in. So retrieving a memory can also make it kind of change over time. And reconsolidation is not necessarily a bad thing. It's probably helpful to have a function that lets us update our representation of things with new information. The consolidation process, it doesn't etch the connections in stone, right? So reactivating the neural connections can strengthen, but also can change some of them, adding new context, or on the downside, adding new errors or bias. So some interesting evidence of reconsolidation actually comes from a behavioral neuroscience study with rats that happened a while back. So there are drugs that we know we can inject which block consolidation, making it so that memories just don't get laid down in a lasting state. So we've got this drug that can block consolidation, and in a rat study back in 2000, they came up with a clever experimental design utilizing just such a drug. So here's their setup. First off, we have condition 1, the first group of rats. They randomly assigned a bunch of rats to these three groups. For this first condition, first group, on day 1, they received some learning, some simple, standard, classical conditioning of the same type that Pavlov had studied. So the researchers played a specific tone, and then always right after that tone, they shocked the rat. They did this over and over and over again to make a clear association between the tone and the shock. This tone means I'm about to feel pain. Something very easy for a rat or most other animals to learn. So at this point on day 1, if we just play the tone by itself, even without a shock, by the end of the day here, they have developed a fear response to the tone. Then those rats, they go sleep. They go to sleep overnight. We come back on day 2, and the researchers inject that consolidation blocking drug. Now, they've already consolidated, right? They already slept on it. They had time to consolidate. It's only on day 2 that we inject this drug. Finally, then we come back on day 3, and let's try something out. What happens if we just play the tone? Do the rats remember that it's scary? In other words, does that memory, did that learning stick around? And the answer is yes. They show a fear response to that tone from back on day 1. So they remember. The information was stored in their brain. That is exactly what we would expect. After all, on the first night, they had a chance for their brain to consolidate all that learning, all that association of the two stimuli. So by the time the drug was injected on day 2, the memory was already permanent, right? Okay, then we come to a second group of rats. We'll call this condition 2. In this group, the rats went through the exact same procedure, the exact same experiences, being shocked the exact same number of times, always after the same tone had played. So their learning experience was identical to condition 1, except in this group, they got the drug that blocks consolidation during day 1. So sleeping that night is not going to help the way it normally would, right? Sure enough, when we come back on day 3 and play the tone, like we did for the rats in the first condition, in these rats, we don't get any fear. The rats in condition 2 have no idea the tone is scary, even though they got shocked with it just as many times. They don't remember that it was associated with shock. The learning didn't get consolidated and made long-lasting. So all of this for condition 1 and 2 here is what we might expect. But it's the third condition in their experiment that's really intriguing here. So for group 3, for these rats, we start out just like in condition 1. They go through the learning where we've paired tone plus shock, tone plus shock, tone plus shock, until they're scared of the tone itself. Then we let them sleep, which means we do let consolidation happen. So their brain has now made a long-lasting, more permanent memory of the tone being scary. But now there's a twist on day 2. We inject the drug that blocks consolidation, which shouldn't matter at this point, because like we saw in condition 1, the drug doesn't matter if you've already slept on it, right? But here we inject the drug and we play the tone, just meaning we reactivate the memory from yesterday. And what's weird is when we get to our test on day 3, just playing the tone by itself to see how they respond, those rats are not afraid of the tone anymore, which is nuts because they definitely went through the same learning as group 1 and they had a chance to consolidate overnight just like group 1. And they got the same amount of the drug at the same time as group 1, but simply reactivating that memory, and in this case, not being able to consolidate it again afterward, seems to have erased or blocked the original memory that had previously been consolidated. So this tells us memory can become fragile again when reactivated. But then it reconsolidates. Usually, right? If you don't have this drug in your system, then the memory will reconsolidate. So that's good. And again, this isn't automatically a bad thing that our brain has this flexibility and can, lose or change memories over time. This mechanism allows us to update our memory to new or changed situations. It allows us to keep learning, like to learn that a tone no longer means I'm going to be shocked, or to learn, that someone with the same name as my ex isn't automatically going to be a jerk, or that loud noises don't always mean great danger for, a veteran who's no longer in war. And in fact, studies going back to the middle 2000s have shown that injecting similar drugs for us humans can undo some unwanted associations in people who have PTSD. So basically you reactivate the memory, but then inject the drug to block consolidation, and then that memory is weakened. So there are applications of this stuff. Now, I'll end this video with an interesting result from 2011. Let's just read through the abstract here. Memory consolidation is a dynamic process. Reconsolidation theory assumes that reactivation during wakefulness transiently destabilizes memories. In other words, makes your memories more fragile again, requiring them to reconsolidate in order to persist. Memory reactivation also occurs during slow wave sleep. We've talked about that, right? And is assumed to underlie the consolidating effect of sleep. Here, in this study, we tested whether the same principle of transient destabilization applies to memory reactivation during slow wave sleep. In other words, they're testing whether activating a memory makes it more fragile and more flexible again, even when that happens during sleeping. So they say, we reactivated memories in humans by presenting associated odor cues, either during slow wave sleep or wakefulness. Basically, it's like the rat study that we just saw, but instead of associations of a tone with a shock, this one involved associations of an odor with something. And here's what they found. As we expected, reactivating during waking destabilized memories, just like with the rat study. In contrast, though, reactivation during slow wave sleep immediately stabilized memories, thereby directly increasing their resistance to interference. In other words, activating a memory while you're sleeping, it helps consolidate the memory. Activating a memory while you're awake, it can make the memory kind of fragile and labile again, so it needs to be reconsolidated once more, and during that process, it may introduce some memory errors. So now you see why I mentioned that sleep is super important for consolidation. It's a way of really laying down some permanence for your memories without the downside of risking them shifting a little bit under your feet. At any rate, that's a good place to end for this video on some of the basic biological mechanisms of memory without getting too lost in the neurochemical details. In the next video, we're going to talk about amnesia and what it tells us about the biological basis of memory.